the Institute of Biological Interfaces at the Kaster Institute for Technology. And her research focuses mainly in single-cell omics. So welcome and thank you for sharing your work with us.
Yeah, thank you very much, Camilla, for inviting me to give the seminar today. If you have any questions during my talk, please feel free to interrupt me here, but there will also be time at the end for you to ask questions. So today I would like to introduce the technique of single-cell omics to you, because this is the major research focus we have in my lab. So far, there are only 3 prokaryotic single-cell pipelines existing worldwide. 2 of them are in the US, one at JGI, Joint Genome Institute in Walnut Creek, California, and the other one is at Bigelow Laboratory in Maine, and the third one is actually located at KIT. I'm going to give you a brief introduction why we do what we do. So we are interested in prokaryotic organisms, so archaea and bacteria. And if you look at the Earth's biomass distribution, you will see that microorganisms actually make up half of Earth's biomass with an estimation of 5 times 10 to the 13 cells. So plants, of course, make up the other half. And here in the middle, the tiny red spot, those are the animals which we actually also belong to. So this is us here in the middle. But microorganisms and plants actually make up the majority of Earth's biomass. And if you look at the distribution of the microbial biomass, you will see that approximately 2% are found in the open ocean, 38% are in terrestrial environments, and the majority, almost 2/3, are actually buried in deep sea sediments. And this is quite interesting, because this is also the reason why we don't know so much about prokaryotic species. So this is just a very simplified tree of life you'll see here. On the right side in green, you see the eukaryotic species. Here, animals, again, this is us, with an estimation of approximately 10 million species inhabiting this planet. 2 million, approximately 2 million are characterized of these eukaryotics. But when you look at the left side of the tree, so in blue, the bacteria, and here the archaea, we don't even know how many species we have. It's certainly not less than the eukaryotic species. It's probably more. So some estimates even say that there are a trillion of prokaryotic species inhabiting this planet. But when you look at the number below here, we so far only have approximately 100,000 of those prokaryotic species described. And this is kind of sad news, but also good news, because that means that we will not get unemployed over the past couple of years or even decades as microbiologists. So why is that there are so little prokaryotic species described in comparison to the eukaryotes? Well, the most obvious thing is, of course, they are really, really tiny, and they almost all look the same, and you definitely need a microscope to look at them. But they are also not very distinct from each other. So what we know is that they're round, cocky-shaped organisms, that they're rod-shaped organisms. But just from looking at an organism, you cannot really tell, is this, an E. coli or is this a salmonella? Whereas when you look at an animal like an elephant, oh, it has big ears, it has a long nose, it's certainly not a giraffe, it's an elephant. So it's far easier to characterize eukaryotes than prokaryotes. And usually if you want to really characterize a prokaryotic species is that you need a culture, you need an isolate of this organism. However, what we know today is that there are less than 1% of prokaryotic species actually cultured in the lab, and over 99% are uncultured. And this is also what we call the so-called microbial dark matter. So this is really organisms that we haven't cultured, that we don't know if we will ever be able to culture them. But the fact is that we were so far not able to get them as an isolate, or at least an enrichment in an axenic culture in the lab, either on plate or in liquid media. And the reasons for that are actually manifold. So one of the main reasons is certainly that microorganisms don't like to live alone. They like to live in communities where they exchange secondary metabolites, where they exchange cofactors, where they exchange nutrients. And usually we don't know what the electron donor or the electron acceptor of an organism is when we know nothing about it. So it's really hard to take an organism out of its natural environment and then find the exact right conditions to culture it. But even if we do, the organism might just grow really slowly, yeah, especially when you think of, I said 60% of the prokaryotic biomass is actually buried in deep sea sediments. And those are actually really slow growing species. They don't have a lot of nutrients down there. They have doubling times of months or even years. They mainly maintain down there, but they're not like our, pet organism E. coli that has doubling times of 20 minutes. They're just really, really slow. And usually a PhD thesis lasts about 3 to 4 years in Germany. So if you have a doubling time of 1 year, this is actually fairly hard to achieve during the course of a PhD. The other reason is that maybe you have an organism that grows fast, but still grows to super low biomass, You don't, if you have an organism that's very efficient in what it's doing, it doesn't have to grow to an optical density of 10, yeah? It might very well be that you just can reach a maximum optical density of 0.01. And then, of course, it's also very hard to cultivate this organism in the lab and actually do something with it. And the fourth and also really problematic reason is that even if you manage to grow something in the lab, it might not show the properties it would show in the environment. A very, very prominent example is, for example, secondary metabolite production as a defense mechanism against other organisms, either prokaryotes, but also eukaryotic species like fungi, for example. So if this organism lives in its environment, and is actually in a competitive situation, it might do other things than it would do than if you grow it in a lab where it has no stress and all the nutrients you need. So this is basically the reason why we're looking at so much microbial dark matter out there. And this is kind of a pity because we know that we can use prokaryotic species for biotechnological applications. Yeah, you all know the use of microorganisms for fermentation, so making alcohol, other platform chemicals that are needed in industry. But we can also use them to make novel natural products, for example, antibiotics. Another big field of biotechnological research for microorganisms is the so-called bioremediation of anthropogenic contaminants, for example, vinyl chlorides, etc. So basically chemicals that were introduced by human beings into the environment and where you can use microorganisms to make harmful products out of it. But it also became the focus of bioenergy production for mobility concepts. And if you think of the e-mobility, we're now all pushing for our mobility concept. You need those very rare earth elements, you need cobalt, you need nickel, and you certainly do not want to wash those compounds out of the soil using like harmful chemicals like acids, etc. So biomining is also a huge biotechnological research field for microorganisms at the moment. But we can only do that and use those microorganisms if we actually have characterized them, if we know what they are doing and if we know how they are doing it, and also if we know how we can manipulate those organisms. So the good thing is that the cost for sequencing, nucleic acid sequencing, has dramatically dropped over the past year. So you see here in 2007, this is where the Illumina technology was introduced. And from there, it went really downwards. And now we have a really low price for a base pair of DNA sequencing. This was, of course, not driven by microbiology. This was, of course, driven by the Human Microbiome Project, by the Human Genome Project. where the goal was to actually sequence a human being, so whole human being, for $1. So that was basically the driving force behind that. But of course, also microbiologists made use of the massive drop in cost for sequencing over the past years. And you can see here two phylogenetic trees of life. One was published in Science in 1997, and this is a phylogenetic tree that was published by my friend Laura Huck in 2017 in Nature. And what you actually see is that there is a massive explosion of prokaryotic phyla. So here we have the eukaryotes, and here we have the bacteria, here we have the archaea, where we actually had no clue that they existed before. And one branch of the tree here is the so-called candidate phyla radiation, or CPR. And you can see all the phyla in this branch that actually have a red dot. Those are all phyla where we have no isolates in the lab. We just have a 16S sequence or a marker gene sequence. We know that they represent something new, but we have actually no real culture, no isolate, no exenic or enrichment culture in the lab. And this candidate phyla radiation, you can see that here it makes up a huge proportion of the tree of life. There are novel estimates that they even make up 75% of the whole prokaryotic phyla distribution. And we know they are out there, but basically we have no idea what they are doing or how we can use them. I mean, at least we're trying to get a glimpse of what they are doing and how we can use them. So when you saw the title of my talk as single cell omics, so when we talk about omics, there are actually different types of omics. There's genomics, where you look at the DNA, the genome sequence. There's transcriptomics, where you look at the RNA, so expression profiles, how much a gene is expressed and how much protein is made. Then we have proteomics, where you can look at the different proteins, and metabolomics. And in my talk, I will mainly focus on the genomics part here. And one way of actually getting novel genomes from those uncultured species is by doing so-called metagenome-assembled genomes. And they are actually a very good way of getting an idea what's going on in a certain environment. So what you do here, you probably know that, is you take a sample, it can be any sample, and you extract all the DNA from the sample. And then you sequence, you actually chop the DNA in little pieces and you sequence it. And then in the end, you can assemble and bind together so-called MAGs, Metagenome-Assembled Genomes. And in an ideal case scenario, when you have a sample, then you basically have different MAGs that contribute to different species. The problem with metagenomics, although it works quite nicely, is, however, that depending on the microbial community composition, depending on the abundance of certain organisms, you do not get a nice MAG that really belongs to one species, but you rather have chimeric MAGs. For example, you have MAGs that are composed of different parts of different genomes. And it is known today that most of the time you are not able to actually assemble and bin the genome of a specific species, but you rather have consensus genomes, which are consistent of parts of different organisms that are very, very much alike. Yeah? And the binning algorithms, assembly algorithms, have gotten much better over the past years. But it's still really challenging to really, get fairly complete, good, non-contaminated genomes from one species. You rather end up with consensus genomes. And depending on the biological question you are asking, that's totally fine. You can argue, okay, I do not need to have the the exact genome of this specific species. I rather want to know what's going on there in terms of metabolism. Even if different organisms are doing that, that's fine. But this is something to really keep in mind, that having a MAC does not mean that you have an equivalent to the genome of an isolate. Yeah. The other big problem, and this also is the same problem we have with the algorithms, is that you can absolutely not trace any horizontal gene transfer events. And you can also, when you have plasmids, you can never really say, okay, this really belongs to this species, because the algorithms cannot distinguish if this is really a contamination that came in from another species, or if this really belongs to the genome. And this is just a problem in metagenomics. And another thing I want to point out is that if you have minority community members, so if you have a very complex sample, let's say a soil sample with like billions of species in there, and some species are really in a super low abundance, for example, less than 1% or 1%, you would need like a huge sequencing coverage in order to get a MAC of the species, but very often it's not possible at all. Or you need like a lot of different metagenomes taken at different time points where you can do covariant binning. But again, this is getting really expensive, and it's almost impossible to trace minority community members. And this is really a shame, because sometimes people argue, well, if it's there in such a low abundance, then it's probably not making a huge contribution to the habitat. But this is actually not true, because you can have species that have a very low abundance but have a very high affinity for a certain substrate, so that the enzymes just work super effectively, which means that they don't have to be there in a high abundance. Just with their little abundance, they can already have so much activity, because their enzyme affinity is so high that they have a huge impact on, for example, biochemical cycles in the habitat. So looking at minority community members is very important, and they are especially hard to trace, because those are also the ones that will never grow in the lab, right? When you have a sample and you start getting isolates, of course, whatever is most abundant will always, have an advantage over things that are not so abundant. So this is the problem with MACs. And this is why we started working a while ago on actually isolating single cells in order to get so-called single amplified genomes. So what you do here is you do not take the whole DNA of a community, so you do not take the sample, extract the DNA. What you do is you isolate the cells, you sort single cells from this environment, and then you lyse them, release the DNA. Then you have to do an amplification of the DNA. I will come to that a little later, because a single prokaryotic cell only contains a few femtograms of DNA, which is currently not enough for downstream processes of getting sequenced. So you have to have an amplification step, and then hopefully you end up with a SAG, a single amplified genome. And this method is really a very nice complement to metagenomics, because it really allows to get the genomes of single species and not just pangenomes, and it also allows you to trace horizontal gene transfer events in plasmids. The downside of the method is that at the moment the genomes are more incomplete. I will also talk about the reasons for that a little later. And it's also very expensive for minority community members, or it used to be expensive for minority community members. I hope that would change that a bit, because as you can imagine, if you have a cell that's only 1% abundant in the environment you want to look at, then statistically it's just less likely that you're going to sort the cell and actually get the genome from that. So I would like to walk you through our single cell genomics pipeline we have established at KIT. And as you can see here, there are a few steps. So the first step A is getting, of course, the sample you're interested in. This can be anything, basically from human gut microbiome to soil to water samples. What you then have to do is you have to label the cell, usually. So give it a fluorescent label. Then you sort the cell, usually in 384 well plates. Then you lyse it. Then you do the amplification. Then you can, this is actually optional, you can do a screening step where you can look for maybe 16S RNA marker gene or other metabolic marker genes if what you sorted is actually what you're interested in, and then you can cherry pick the cells you want to sequence, or you can just sequence everything. That's totally up to you. We usually use Illumina and Oxford Nanopore for that, and then in the end we need to conduct, of course, bioinformatic analysis. So basically look at the reads, do decontamination, do filtering, retrimming, etc., assembly, and then we end up with our single amplified genomes. So in my lab, we basically do everything from the sampling down to molecular biology and bioinformatics. And the sampling is, as I already said, really diverse. So we just started a project in our local vineyards, but we also have forest samples. We have deep sea sediment samples where we, go on cruises and get the cores that are basically drilled in the deep sea sediments. We also had a project where we sampled the biofilm of those kelp algae here. So it's very, very diverse, and you can basically can do any sample you like. The only thing you really have to take into account is that you have to make sure that you preserve your cells, because unlike in metagenomics, where it's not a big deal if the cells lyse during the sampling or transportation process, what you really need for the single cell genomics pipeline is to keep the cells intact. So they need to stay as a whole, and usually we preserve them in glycerol and then we freeze them away in liquid nitrogen, and that helps to keep them intact until we can start the process of single cell genomics on them. So every sample, this is also one of the take-home message, needs a certain type of preservation, and this is sometimes also try and error. So not everything works for every sample. And you usually have to play around with that a little bit, but one thing that's absolutely mandatory and always needed is to have glycerol in your slurry when you stir the samples in order to keep them intact. So the second step of the pipeline is also one of the most important ones, and that is the labeling of the cells. And usually the labeling can be done randomly. So for example, using Cyto9. So that means that you would label every cell in the sample, no matter what. However, this leaves you with a problem that you cannot selectively sort species that are of certain interest to you. So what we have developed over the past years was actually an approach where we did fluorescent in situ hybridization with the cells, and we're then able to sort them based on certain phylogenetic or metabolic marker genes. So you would think that this is pretty straightforward. However, it's not, because the usual FISH process, you might know that, usually requires fixation. So that means that you basically bring the cells on a slide or a filter paper. So this is bad. This is good for microscopy. If you want to look at the cells, that's fine. But if you want to actually have them free flowing to sort them later in those 384 well plates, you need them detached. And the process of detaching them is very painful and actually kills a lot of cells. And then the second big problem is that in order to get the probe, the fluorescent probe inside of your cell, you usually need a type of permeabilization, and this is usually done with formaldehyde, performaldehyde. The problem with paraformaldehyde is that it allows the probe nicely to penetrate the cell, and you get great fluorescent signal, and you can do really good fluorescent in situ hybridization. However, when you use that, you form DNA protein aggregates, and then your DNA will not amplify in the downstream process again. So when you usually in the past did fish with the cells, you could not do any amplification, no PCR, and also no library preparation for sequencing. So that's why we had to come up with a method where we basically omitted the step of fixation and also permeabilization. And we ended up developing a method which we call fixation-free fluorescence in situ hybridization without additional permeabilization. And this was a lot of work and a lot of trial and error. And we published 2 papers on that, but we could nicely show that this technique is really suitable for minority microbial community members. So if you have cells that are less than 1% abundant in your microbial community, you could, for example, first do an amplicon sequencing or metagenomic sequencing, then see what's in there, then specifically design probes for those cells, and then basically do the fixation-free fish, and then sort them, and then you're able to get sacs of those microbial community members. And this does not only work on phylogenetic marker genes, such as the 16S RNA, but also metabolic marker genes. So if you're interested in a certain metabolism where you have a good marker gene, like for example MCRA for methanogenesis, or PMOA for methylotrophs or methanotrophs, then you can specifically pick those genes and do a ring fish or a dope fish, for example, and label the cells for targeted fluorescent sorting. So if you're more interested in how this method works, I recommend checking those 2 papers out. The third step in the pipeline is the sorting. And sorting is usually done via fluorescent activated cell sorting, so FACS. And the process of that is that you have your cells, your labeled cells, and your maybe unlabeled cells if you did a targeted fish. And what happens is that the cells get pressed through a nozzle with high pressure, 8 bar, and then you have a laser beam that's shot on the cell, and then you have a forward scatter and a side scatter that is reflected and detected by the machine, and then basically there is a charge applied to this droplet that contains the cell of interest, which you picked before, and then basically you can sort the ones you want in one well and the ones you don't want in the other well. And we usually use those 384 well plates, and then each of the well contains a single cell. Some, as positive controls, contain more cells, 100 cells, and also some do not contain a cell at all. This is our negative control then. So this is basically the standard thing for the single cell genomics pipeline, using the FACS for that. Yeah, this is really fast, this is really high throughput, and this usually works well. However, when the cells have undergone the FISH procedure and when you have sensitive cells, we often observe that the cells lyse before they actually hit the bottom of the 384 well plate. So remember, I said a few seconds ago that the cells are pressed through this nozzle with 8 bars, so that is really high pressure. So if the cell is already a bit damaged or doesn't have a super strong cell wall, the cell might actually lyse before it can actually be sorted in the 384 well plate. And this is a problem, which is why we came up with a novel sorting method. We're working together with a company called Cytina. They used to be a startup. Now they're not a startup anymore, I guess, because they progressed quite nicely. And this is a sorting method that's not based on those fluorescent activated cell sorting, pressing the cells through a nozzle, but rather a piezoelectronic piston where the cells are gently sucked out this nozzle here. I hope you see my cursor. And then basically also get a deflective charge, and one can detect if it's a single cell or if it's a multiple cell. And the nice thing about this method is, I hope you can see that here, that instead of getting those scattergrams from the forward or the side scatter in the fax, you actually get pictures of how the cells look like. So here you can see the cells. Here are the fluorescent cells. So here you don't see it, then it comes a little closer to the edge of the nozzle, and then you actually know which kind of cell you have sorted. And this works quite nicely. Unfortunately, not as high throughput as the fax machine. So in the fax, you can probably sort like 10 million cells in 20 minutes. With this cell printer here, you can maybe sort 10,000 cells in an hour. So it's not bad, but it requires more sample because the more cells are thrown out, and it's just not as high throughput, but it's much more gently than the fax. And we published a paper in 2020 where we actually took a wastewater sample and looked at single cells from those candidate phyla radiation, and we did it in comparison to fax and the cell printer, and we could recover much more sacs of those candidate phyla radiation with the cell printer, because apparently they had a delicate cell wall and they lysed easily in the fax, and we got a lot of sacs from this candidate phyla radiation and could significantly enhance the tree of life on those different phyla. So this is another paper we published in 2020. If you want to know more, please feel free to check it out. So this is a nice instrument if you have delicate cells that like to lyse easily. After the sorting, the cells need to be lysed. I will tell you about that a little later, because the lysis step is actually also pretty crucial. You don't want to lyse the cells too harshly to destroy those tiny femtogram amounts of DNA and RNA in the cell. Also want to do it harshly enough so that the cell is actually broken open and releases the DNA, and that you can then actually do the amplification. And the amplification process, of course, cannot be PCR based, because when you're looking at unknown organisms, you cannot do PCR because you don't know how the primer should look like. So the method of choice here in the single cell genomics pipeline is the so-called multiple displacement amplification. And it works with a specific Phi29 polymerase, which has a high branch displacement activity and is also much more precise than, for example, Taq polymerase. So it has a much lower error frequency. And then what we have instead of primers, we have so-called random hexamers. So you have hexamers with different G, A, T, and C composition. So 4 to the 6 different types. So this is like over 4,000 different combinations. And then, of course, your DNTPs and then your template DNA. And the way this works is that you denature your DNA, and then you random hexamers anneal at the strand where they fit, when they actually have a good fit. And then the polymerase starts making double-stranded DNA. And whenever it hits another random hexamer, so whenever it hits a double-strand, it actually branches off. And this goes on and on and on and on. And this works quite nicely to get nano or even micrograms of DNA from those femtograms of DNA from a single cell. However, the disadvantage of this method is that it's expensive, so the Phi29 is pretty costly. It's biased against high GC regions because the polymerase is temperature sensitive. It's not like a Taq polymerase. You can heat up to 95 degrees Celsius. the reaction actually has to be carried out at 37 degrees Celsius, and we all know GC has 3 hydrogenase bonds and AT only has 2, so you usually would need higher energy to form those 3 between G and C. So this is why it's basically biased against GC, because the annealing is not that good of the hexamers. And that usually leads to those uneven genome amplifications. You remember at the beginning I said the sex are usually more incomplete than the max, and this is part of the reason why this is the case, because we have this bias against the high GC. You also quite often chimeras are formed if you're unlucky. And of course, one problem, which is why you cannot just do it under a usual clean hood, is that it's very easily contaminated. If you work with so small amounts of DNA, you basically have to work under forensic conditions in order not to amplify your human genome, or whatever is flying around in the lab. Yeah, so we have an ISO 4 clean room in my institute where we can perform those MDAs under really contaminated frequent not contained cell, because if also amplifying then in there, then you can take your plate and open your little bucket and dump it in and close it, because that means that everything's contaminated. So this is the problem with the MDA, and this also causes the problem that when you want to do a PCR screening after the MDA for specific marker genes, like for example the 16S RNA, if you were unlucky, then this is just the marker gene that did not amplify. Yeah, and then you screen and you think, oh, this is not my cell of interest, but it's actually only because this region did not get amplified and your cell would be in there. So this is a problem, and this is also why we end up often with having incomplete genomes. Now, another reason which I failed to mention, which has not directly to do with the MDA, which is a general problem when you work in super small volumes with super small amounts, is that when you have this DNA in your well, in your 384 well, and it anneals to the wall of the well, or it forms a loop structure. Sometimes when you denature, DNA is not necessarily like totally linear, but it likes to make those little loops. Then all of a sudden you have double-stranded DNA anymore, and then your hexamers can't anneal, your polymerase cannot anneal. That means that this region does not get amplified. This is also a reason why you end up with incomplete genomes, because you basically have some stereo for the hexamers and for the polymerase to anneal. Yeah, so this is also something to keep in mind. And this is actually an average success of an 384 well plate from a deep sea sediment sample. And I'm showing you that to show you that the success is actually pretty crappy. And part of the reason is actually the lysis, because when you work with samples from the deep sea, you can imagine that the cells that are living down there are pretty tough cookies. Yeah, they have to withstand like really, really high pressure. They're several hundred, if not 1,000 meters below seafloor. So they need to have a really tough cell wall in order to withstand those harsh pressures and those harsh conditions. And so it's not that easy to crack those cells open and to lyse those cells. And lysis is really crucial for the success of the MDA and the success of single-cell genomics in general. So whenever you work with a sample, you always have to test out lysis condition. There is not one lysis condition that would work with every sample. Of course, you start with like a general lysis, but then usually you really have to look at your MDA success and then start to modify your lysis condition in order to, be able to crack more cells open. The same thing goes the other way around. Don't be too harsh. If you have cells that are basically already lysing when they hit the bottom of the 384 well plate, basically if you would jump into a pool with very little water and you would hit the concrete, then you would probably also lyse, and this is also what happens with the cells. And when you then add some harsh potassium hydroxide or some lysozyme, this will damage the DNA, and then of course the MDA can also not work. So lysis is very crucial in single cell genomics. So this is another take-home message I want you to be aware of. And since this is such a huge problem, and since the costs and the success of the whole single cell genomics pipeline really rely on the amplification, we are doing a lot to actually improve the MDA. And one way of doing it is that we are drastically reducing the volumes from microliter to nanoliters. And this is not only reducing the cost of the reaction, but you can also imagine that basically when you have a smaller volume, which is of course more difficult to work with, but then basically the components in your solution also have statistically a higher chance of finding themselves. If you add a drop of color in an ocean water, then it will really dilute out, and when you add a drop of red and a drop of blue, then it will never basically meet to form a lilac color. If you do it in a very small volume of a small schnapps glass, basically, then the molecules will have a statistically much higher chance of meeting. So volume reduction is one way of improving the MDA. And the latest method we're testing and we're looking at is a so-called Droplet microarray platform, which is consistent of hydrophilic spots with a superhydrophilic barrier. And the nice thing about this is that instead of this 384 well plate, you basically sort your cells into those spots, and they are separated, so they won't mix. So you can really get your single cells in there, but you have a much smaller volume. And in contrast to the 384 well plate, where you basically have this conical shape of your plastic, and where the DNA has a chance to stick to basically everywhere here on this plastic, here you just have one surface where the DNA can stick to, and the rest is basically the droplet. So this is also decreasing the chance of the DNA to basically stick to the plastic. And we did some tests on that. The paper is currently submitted. It's not out yet, where we took different volumes. So please keep in mind that this is the total reaction volume, also with the sorting liquid and with the lysis and everything. But we could actually show that by reducing the volume 10 times, we get much less contamination, we get much less duplicates, we get a much higher mapping coverage. So this was done with E. coli single cells, and a much more equal redistribution, and this ultimately leads to more complete sacs. So just to give you a few numbers, the average sac genome recovery rate for an E. coli cell before our work was approximately 30%. So 30% of the genome got recovered in the sac, which is kind of really sad if you think that 70% of the genome basically remain in the complete darkness, because you have the MDA bias and problems going on during the amplification. And with those reduced volumes, we could actually increase the genome coverage to 80%. So 80% is actually something you can work with, and also something where you can reconstruct metabolism and then say, okay, we're fairly confident that the genome of the cell looks like that, and this is what the cell is doing in the environment. So this actually worked really well. The other thing I'm not going to talk about, because as far as I could see, John Falmos, my bioinformatics group leader, also already gave a talk in the seminar series, is our bioinformatics pipeline development. So this is what we're also doing. We're trying to improve assembly and binning algorithms, and also to basically help cleaning up genomes of microbial cells from contamination. And what he developed, and there was a paper that was published in Nucleic Acid Research this year, is the so-called MDM cleaner, which is a pipeline that actually allows you not only for SACs but also for MACs to nicely decontaminate the cells and basically get much purer MACs and SACs out of it, although they might be a little less complete, but then you can be really certain that what you see in those genomes is really what is there, yeah, and not some random contact that got binned into the same MAC or SAC. Okay, so with that, I would like to conclude that with omics, especially single-cell genomics, with those culture-independent methods, we could significantly increase the number of genomes we have for the phyla out there. I mean, you see that some, like the really common well-growing organisms like the gamma proteobacteria, you basically don't need any MACs or SACs here because there are so many isolates existing, yeah, you see. But with like the candidate phyla radiation here, we only have MACs and SACs to rely on to see what they are doing. And you can see that the numbers of culture-independent genomes is drastically increasing with the drops of sequencing costs. Yeah, and this is all nice, and this is all really important because we need to know what's out there. But what microbial ecologists typically also want to know, except who's in there and basically what are they doing is also how are they doing it and to what extent are they doing it. And this is a question you cannot really answer with genomics. This is where you actually need to look at transcriptomics. And over the past 2 years, we also have developed a prokaryotic single-cell transcriptomic pipeline in our lab. This is also work that's recently in review. My PhD student, Morgan Sobol. And the reason why this is important is that when you look at microbial community in a bulk analysis, you only get the average gene expression from all cells. Yeah, you basically, the cellular heterogeneity is like completely masked. And this can be especially problematic when looking at, specific responses, also in minority community members, for example, when looking for secondary metabolites, if like all the other genes are like so expressed and your little secondary metabolite producer is a minority member, then you will just not see the expression because it's masked from all the other cells. And this is a problem that we can solve actually with single cells, because there each cell type gets a distinct expression profile, and you can really reveal the heterogeneity and subpopulation expression variability of the cells. And this is also specifically important when you, for example, look at biofilm formation, because the cells that are already in the biofilm have vastly different expression patterns than the cells that are swimming towards the biofilm or are at the surface of the biofilm. Yeah, they are doing different things, but if you would just scoop up the biofilm and do a total RNA metatranscriptome, you would never see those differences. And the reason why single-cell RNA sequencing, single-cell transcriptomics is quite difficult, much more difficult than genomics, is basically shown in this little graph here. So in black, you see the eukaryotic single-cell transcriptomic methods, and in red, the prokaryotic transcriptomic methods. And you see that there are some here popping up over the years. However, they all rely on microfluidics and are not really easily reproducible. And one of the reasons is because you need those specialized microfluidic devices, which you cannot just order online. You basically need a facility that designs and makes the chips for you. And interestingly, besides from the original publications, none of those methods, so PeterSpec or microsprit-sec, they have never been used in other studies and reproduced by other labs, because it's very, very specialized in terms of those microfluidic devices, what you need. And you can see that there are, however, a lot of methods for eukaryotic species, and they work very well, and they got reproduced. And the main reason for that is when you have eukaryotic cells, in this case a mammalian cell, you have, of course, much more RNA in there. Yeah, that's a larger cell, it's just more RNA. That's just yeah, very logical. But also, one thing that distinguishes the eukaryotic cell from the prokaryotic cell is that here the mRNA is polyadenylated, which means that you can easily deplete it. Yeah, you can easily distinguish your mRNA from the rest of the RNA. And another big plus is that the half-life of the mRNA is approximately 10 hours, whereas the half-life of the mRNA in a prokaryotic cell is approximately 5 minutes. So you can imagine that you have very little time of getting your single-cell transcriptomics to work in a prokaryotic cell. And the third reason is that you have really diverse cell wall structures. This is what I said at the beginning. You know, a deep sea sediment cell is much tougher to lyse than another cell. But if you don't know what you're looking at, then it's really hard to find out what's best for the cell and what's best for your single cell transcriptomics. And of course, a big problem which holds true for both eukaryotic and prokaryotic cells is that most of the cell's total RNA is, of course, not mRNA, but rRNA. So having a way to actually, you know, specifically target the mRNA by this polyadenylation is actually really great. Whereas here in the prokaryotic cell, it's just all, you know, basically all together, and most of it is the rRNA. Nevertheless, we found a workaround and established a single cell transcriptomic pipeline. And the key step was actually in the lysis and amplification. So we found a way to remove the gDNA, do a reverse transcription, a ligation, and then perform multiple displacement amplification. And this was not an easy process, and the key step of it was being like very, very fast and doing all within, yeah, I would say basically taking the sample, sorting it, doing it all within like 30 minutes. If you let the sample sit for 5 minutes too long on the bench because you need to grab a bite or need to run to the restroom, then basically you will not get a single cell transcriptome out of it because your mRNA is degraded. So speed is key here, but nevertheless, we managed, and we performed a nice experiment, which I'd like to show you. Also to show you that heterogeneity of cells is really important. And what we took was Escherichia coli as a model organism. And we took a culture which we split up, and we heat shocked one part, and the other one we didn't heat shock. And then we did differential expression analysis. So we, went through the single cell pipeline and sequenced the cells, and we had 1, 10, 100, and bulk mRNA heat shock and non-heat shock treated. And what you can nicely see is that you have a really heterogeneous expression profile pattern among those single cells, whereas when you're looking at the bulk samples, it's all just one big mess, and you cannot distinguish anything, basically. And this was actually confirmed by doing a pseudo bulk analysis. So what we did is we took the 1, the 10, and the 100 cells together and the bulk samples together, and you can see that those clustered like really nicely down below here, the heat shock ones, and up here the non-treated ones. So this is actually a very nice experiment to show that heterogeneity is in fact an issue and that it's important to look at it. And here we specifically looked at the heat shock gene count because, you know, in E. coli we know what they are. And you can also see that we have a nicely distinguished pattern between the heat shocked and the non-treated cells when we look at the different amounts of cells. And here this is for 120 most significant genes. And I'm kind of running out of time here, so you just have to believe me that single-cell transcriptomics is important to actually capture the cell's heterogeneity, and that a lot of things are masked when you just look at the bulk metatranscriptomes, and that this is important for certain questions, especially when it comes to finding, for example, natural product producers that are not super abundant in an environment. You really want to be able to capture those and distinguish the expression profiles from the rest of the cells that are inhabited. Okay, with that, I would like to conclude. So we are really out there to hunt the microbial dark matter and to shed light onto microbial dark matter. And I want to end with a quote of Victor Hugo from Les Miserables, where the telescope ends, the microscope begins. Which of the two has the grander view? I guess that's to us to answer. This is my group. You can also see Camilla here. Where's Camilla? There, right next to me. So she's been a visiting postdoc in my lab for a couple of months. We're really happy that we have her, and I'm still collaborating with her. And thank you so much for letting me give the seminar, and I hope you have a lot of questions.
Thank you, Anna. Is there any questions?
Congratulations, and thank you for participating in our seminars. They are really interesting results. I think that they can be an important advance for the bacterial universe. I have one or two questions. I hope, yes, we hope so. I have one or two questions. What bacterial populations are you focusing on to do this type of single-cell studies? And the second question was if you have worked with strains resistance to antibiotics.
Yes. I'll show I've prepared a backup slide for that. We are interested in certainly novel natural product producers, for example, from algal biofilms of kelp forest. We also have projects where we are looking for novel bioremediating bacteria in soils, but also in river sediments from industrial sites. We are very interested in extremophiles, especially chloroflexi and archaea from hot springs. So we have a huge hot spring project going on at the moment with hot springs from over 17 different countries, where we're looking especially at chloroflexi. I'm an archaea person by heart, so and archaea are notoriously understudied, so those are also a big focus of our work. We have a lot of marine subsurface samples back from my time at Stanford University, and those are notoriously hard to cultivate, especially because they are anaerobic and live in very oligotrophic environments. So we have a lot of we have a whole -80 freezer full of deep sea sediment samples, but we are also very interested in methanogens, methanotrophs, methylotrophs, so generally organisms that influence the carbon cycles, especially the methane cycle. And one project we recently finished was that we labeled the PMA gene of a previously unknown methanotroph called USA alpha, which was also a minority community member in forest soils. And where people have tried to cultivate it for over 20 years and also to try to get the 16S RNA sequence, but never succeeded. And we're actually able to get single amplified genomes and also the 16S RNA sequence by fluorescently labeling this PMOA marker gene. And then we finally had a 16S sequence, and we finally had a fairly complete genome. And it turned out that this organism had another interesting gene in there, XOXF, which was known from other organism earth element as cofactor. Naturally, nobody has ever tried to cultivate this organism with lanthanide in the media, but from the omics data, we now knew that it needed lanthanide as a rare earth element, and we cultured it with lanthanide, and suddenly it was growing. And, yeah, now there's a fairly clean isolate we have of that. So this is also an example where basically getting the genomic information first helps to do the cultivation later. And Jochen Müller, who Camilla also knows really well, joined my lab beginning of the year as group leader, and he is focusing on antibiotic resistance. Yes. And has a lot of projects going on with antibiotic resistance. And this is also a topic which is, I think, really well suited for single cell genomics, because you know that those antibiotic resistant genes are often transferred via horizontal gene transfer. You can often find them on plasmids, basically, and this is where metagenomics is not very suited to basically trace the evolution and the spread of antibiotic resistant genes. And this is where the single cell genomics also comes in very handy. So we're really happy that we have him in our lab now, and he's focusing on the
of course. They are amazing subjects. And do you think that it's possible to evaluate the rate of horizontal genetic transfer, for example? I think that is possible.
Yeah,
it's
certainly.
Possible.
If you have enough, you can do it.
Sorry. Thank you.
Okay. I think it's possible. You just need, of course, enough stacks for doing that. Yeah, it needs to be high throughput. I mean, that's the case for many biological questions, and that's also a critique we get very often. Like, you know, how do you want to evaluate an environment when you just look at 100 cells, 100 single cells? And that's totally valid, this argument. And really, the ultimate goal is not to only look at 100 or 1,000, but basically to look at the million cells that are in there to piece together the evolutionary relationships and what's going on in their environment. But we can only do that if the method gets cheaper. You know, if you have to pay €5 for an MDA reaction, you cannot look at 1 million cells, right? We need to decrease the costs, and I think we're on the right path of doing that, but it's still a long way to go.
That's very interesting, and thank you very much. Thank you.
Are there any other questions? I have a methodological question. Before doing the sorting, you label the cells with probes, right? But how do you design this probe if you don't know what you're looking for?
Well, we usually first, so when we're looking just for a specific taxa, it's usually 16S RNA, which we know from amplicon sequencing, basically, or metagenomics. If we're looking for a specific metabolism, then we do a metagenome. So this is usually standard procedure to do metagenomics before, also to know that when we have the sample, that what we are looking for is actually still in the sample. Because when you have like a sample and then you have an old metagenome and you're like, oh, this is interesting, maybe we should look for, you know, something specific in there, and then you go back 2 weeks later and take the sample from the same spot, but the community might have shifted already. Yeah, so we always do a metagenome from the same sample we do the single cell genomics with. And yeah, and I'm not saying metagenomics is a bad method. I'm saying it's very complementary to single cell genomics, and you can use the information of both to actually help answering your biological question. And as for the probe design, this usually comes from the metagenome then, because if we see the gene is in there, then we know, okay, there's something we can label.
Thank you. And then I had like a more general question. Is there at the moment like a question that you really would like to answer with this methodology, like the question that you are the most interested in?
Yeah, so my lab is really focused on methodological development to basically get everything cheaper and better and more high throughput and faster, and get everything automized with liquid handlers and stuff like that. This is also because I did my PhD on them, but I'm mostly interested in archaea because I think they have the most fascinating metabolism, and what we are currently, so this is currently a new project we started, is that we actually want to decipher hypothetical proteins, so proteins that do not have a function, which are sometimes like 80, when you look at an archaeal genome, sometimes you have like 80% of the protein encoding genes have actually no assigned function. There's no biochemistry, there's no structure, no nothing. So we want to use the single cell transcriptomics method and also deep learning algorithms to try to shed light on those hypothetical proteins. So that's my pet project at the moment, I say.
Okay, thank you. Well, I think it was a very complete presentation, and it was very clear, and I learned a lot today, and I hope other people too. And yeah, if there are no other questions left, we say goodbye till the next time.
Yeah, thank you so much. And if you have any more questions, which might come up later, I think I had it on my first slide. I had my email address, so
thank you.
Feel free to shoot me an email with questions. And
thank you very much, Anne. Thank you. Bye bye. Thank you very much.
Bye, guys. Bye.